today will be. It's a great pleasure to introduce the speaker for today, Dr. Greg Kaczorowski. I've known Greg 40-some years now. And so I'm used to him telling me that he was born, raised, and educated in South Bend, Indiana. So from that information, you could tell he did his undergraduate at the University of Notre Dame, where he graduated in 1972 with a major in chemistry, and he graduated summa cum laude from Notre Dame. He then went on to MIT, where he was, I think, Chris Walsh's first graduate student, or one of the first students, number one, he said. And he received a PhD from MIT in biochemistry. From there, he went to the Roche Institute for molecular biology, where he did a postdoctoral study. And more importantly, he met his wife there, Dr. Maria Garcia, who was educated in Spain and was also doing a postdoc at the Roche Institute. And in 1980, both of them were hired by Merck, and with the sole purpose of establishing a department of ion channels, which Gray led until his retirement in 2009. He currently has a consulting business with his wife, and I'm not sure who's the CEO of that group, but no, Maria, she is. So, Gray has, I think, over 225 peer-reviewed publications. And perhaps more importantly, if you have any interest in model railroads, you might want to talk to Greg after the end of this presentation, because he is a trained fanatic. When he first joined Merck, him and another colleague used to go and camp out on the border between Maryland and central Pennsylvania just to watch the trains go by. So with no further ado, I'd like to introduce Greg.
All right. So thank you, Matt, who I've known for a long time. I gather by looking at the audience, since you're my peer group, you don't know what an ion channel is, or if you do, you don't know anything about the particulars of it, because the concept really did not exist when I started in science. And it's not something I wanted to do at all. So before I give the talk, just by a quick story. By the way, I've had a lot of postdocs and scientific colleagues in Austria. And I've been in a hotel overlooking that scene from The Sound of Music where that was filmed. And those songs are very sentimental for me, because a lot of my friends live there. This is completely by accident that I got into this field. I mean, I was trained like Matt as a chemist, and I wanted to do biochemistry. And what people did in those days, they studied enzymes. These are proteins that synthesize or degrade various molecules quickly. And I had no thought of doing what I'm going to talk about today. And it wasn't even described. When I was lucky enough to, well, join Chris Walsh's lab, he trained over 500 people doing enzymology, and I'm not one of them. I was his first student from MIT's biology department. He says, I want you to study bacterial membrane transport. And I said, what? I didn't come here to do this. But, you know, I'm a graduate student. He's a professor, although he's like 4 years older than me, and had just come 3 months before me. But he had a colleague at the, and this is a project he was supposed to do when he was a postdoc, and he didn't do it, so I did. So he had a colleague at the Roche Institute of Molecular Biology, Ron Kabak, who turned out to be a really strong influence in my life. He's Mr. Transport. He's the one who really founded the field of membrane transport. And that's what I did as a graduate student with Ron's help. And then I went and did a postdoc with Ron and learned all about transport proteins and met my wife as Dr. Garcia. as Matt mentioned. And then I'm looking for a job, and I'm thinking, well, you know, membrane transporters are very important for drug discovery and development, because neurotransmitter blockers, uptake blockers are used. Farxiga, you know what Farxiga is? Right, on television, this SGLT2 transport inhibitor blocks glucose uptake from the kidney. You pee out the glucose. It has great cardiovascular properties too, for congestive heart failure and kidney failure. There are drugs on transporters, but I'm clever, right? So I said, I'm going to go and work on transporters and these new proteins that haven't been described yet, called ion channels. And I went out and gave an academic talk, and they laughed at me. They said, you don't know anything about, nobody knows anything about this, and you're going to come here, you're never going to get a grant. They don't fund you. The government doesn't fund you to do things new. You're supposed to work on things that you know how to do, to have incremental increases in knowledge. And I was really crestfallen because, first of all, I thought that was a pretty clever thing to do. Work on ion channels. What am I going to do? It just so happens one of my friends was at Merck, and he said, they're hiring biochemists, so you should go and give a talk. And I did. And then I got to meet Roy Vagelos, who was president of the research laboratories, very famous scientist. And I had a meeting with him, and I said, I told him what I wanted to do, and I was ready. I thought he's going to laugh at me and throw me out of his office. And he said, we'd be happy to have you come and do this. And if you're successful, you could have a department in 5 years. And I said, no way. It took me 3 months to say yes, because I just didn't believe him. But he didn't tell me that Merck had lost the ability to license one of the first very important ion channel modulators, nifedipine, which is lowers blood pressure, because people had told him that would be a toxic drug. More importantly, they had a drug for animal health, ivermectin, which we'll talk about, which nobody knew how it worked, but it was important for animal health and had applications in human health. And so that it's an ion channel modulator. Okay, so going to the right place at the right time stupidly and telling people what you want to do, sometimes they say it's okay. And that's the story of how I started. So I'm going to be talking about this target class. And you notice 15 years after retiring from Merck, Maria and I started Kanalis Consulting. Kanalis is the Greek word for channel. That's named in honor of Roy Vagelos. It's part of his legacy, channel consulting, because he allowed me to work on this. And Dr. Maria Garcia teaches a course at Drew University for entry-level medicinal chemists. And she talks about this target class. So I borrowed exhaustively from her slides. She teaches a 90-minute course with 60 slides, and she tells me when they walk out of there, they look like deer in the headlights. So we're not going to get into details. We're just going to do a 10,000. I think somebody who studies ion channels would figure that out. But, all right, so I'm going to give you an introduction about what these things look like and what they do in general. Then we're going to hopefully convince you that they control all of cell physiology in the body. Your body works off electricity, and this is the mechanism by which electricity is signaled and transduced in the body. We're going to look quickly at the structure of these proteins. Ion channel modulators are very important. That's where your drugs come from, all right? But that's how a lot of organisms like scorpions and spiders kill you, because they make ion channel blockers, and they do their job. That's how they paralyze their prey. They also, from Matt's point of view, many medicinal chemists make drugs on other targets, and they don't know why they have toxicity sometimes in safety. It's because they inadvertently target ion channels in the heart, for example, or the brain. So they're not only targets for therapy, they're targets for safety. And then finally, I'll finish with how to make an ion channel drug, which actually, I'm talking about the field now, okay? Because I started when they didn't know anything about it, and it was a lot of academic people and about 4 Nobel Prizes that allowed this to actually take place. And I'm very proud that it took place for the first time at Merck. So If you don't know anything about ion channels and you leave here, just remember, an ion channel is a hole with a physiological role. This is a start of a poem written by an ion channelologist back in the 80s for his colleagues who said, what are you working on? All right. So actually, in the whole 20th century, the physiology studies of the 20th century, we described the work of ion channels and how the heart works, how we breathe, muscle contraction, how we secrete hormones, how we think. All right. That's general physiology, but the basis for this was not known. So around 1980, when I got into the business, we had the ability, because these are membrane proteins. Nobody purified membrane proteins. They're stuck in lipids. They're in grease. You have to know about detergents. You have to pull these things out in an active form. That was unknown when I started. And really, the field helped us be able to do that. And then finally, I'll talk about molecular pharmacology, which is the science of making drugs. You all remember what a cell is, I hope, from high school biology? All right. So the cell has a plasma membrane that surrounds it. And Peter Mitchell got a Nobel Prize in 1978 for a unified theory of bioenergetics. So the cell, in order to function, uses the energy source ATP to pump ions out of the cell. For example, the sodium potassium ATPase. It pumps, I mean, well, you live in an environment which is high in sodium, low in potassium. And so the pump, by moving 3 ions in one direction, plus charge, and 2 in the other direction, sets up a potential across the cell membrane. That potential is like 80 millivolts. So what does that mean? So 80 millivolts, if you look at a pin, which is 1 millimeter in diameter, that's a 9,000 volt field across 1 millimeter. That's a tremendous electrical field that the proteins sit in, right? They also have the cell pumps ions against their gradient so that sodium is high outside and it's low inside, and potassium's low outside, and it pumps to make it high inside, and it pumps sodium, which is high inside, out. So sodium can move down its concentration gradient into the cell, or potassium can move out of its concentration gradient, energizing the cell, all right? And so you're looking at this and saying, well, what's the flagella? We don't have flagella on our cells. Well, your sperm cells do, and your sperm is actually powered by calcium channel activity, all right? So I had a client wanted to make a contraceptive by blocking that calcium channel, paralyze the sperm. I said, well, you know, you're going to have to block 100% of the channels 100% of the time, because one of those little rascals escapes. You don't have a contraceptive anymore. You would understand that. All right, so overall view, what do these things look like? So at the top right, this is a protein embedded in the membrane. The top is outside, and the bottom of the protein is inside the cell, and it's sitting in an oil bilayer, okay? It's in a tremendous electrical field, so you can make that protein change conformational change and open so ions can move by changing the membrane potential across the cell. Or you can add a ligand like acetylcholine, which binds to some ion channels and causes them to open. You know, nerve gas is cholinesterase inhibitors, okay? So if you have a lot, or Novichok is another one. If you block acetylcholinesterase, there's a lot of acetylcholine that opens the channels and keeps them open, and you die. It's a nerve toxin. So, you know, we figured out how to kill people with ion channels as well as the organisms. So these basically control all types of physiological activity, setting resting potentials of cells, controlling calcium homeostasis in the cell, electrical signaling. Your ability to live outside the ocean. You have kidneys, that's driven by ion channel. Ionic homeostasis in the body is driven by ion channel activity, your ability to move muscles. And basically, this is really a simple idea. If you look at the little drawing here, gating is just the opening and closing of an ion channel. You see what it looks like closed? And then there's a conformational change and it opens so ions can move. And they move incredibly fast. They move at 10 to the 9th. That's like diffusion control. Like you put salt in a glass of water and it dissolves and the ions move at this. It's called diffusion control. It's very fast. Well, that's how fast the ions move through the pore. Okay, so these are for rapid signaling. Transporters, which I also studied, they turn over, or their rate of turnover, 6 orders of magnitude slower, okay, than these proteins. So these are proteins that are very important. They can signal very, very, in the course of milliseconds. And then permeation is the other characteristic. Basically, you have 5 ions that control biological signaling: sodium, potassium, calcium, protons, or hydrogen ions sometimes, and chloride ions. Those are the ions that move through ion channels, and some are non-selective that move everything, monovalence or divalence, but most of the important ones are sodium selective, potassium selective, calcium selective, chloride selective. So that's the basis. Now, what do? They do everything. Your heart beats because of it. You think because of it. You can move your muscle. You secrete hormones, insulin, because of ion channel activity. Your kidney works. Your airway epithelial, your endothelial are all regulating ion movement or water movement with the ions based on ion channel. Pain signaling. When you have pain, it's because your sodium channels are firing and the signal is sent up to the brain. The 5 senses are all based on ion channel activity. Even the immune response system making antibodies because of ion channel activity, and many, many more physiological functions. So I was trying to think about what can I tell you about ion channels that would have some, your own experience would say, oh, that's what's working in my body. Your ability to differentiate Tabasco sauce from horseradish, grated horseradish, from spicy mustard is because of 3 different types of ion channels called trip channels, which are sensed by the active ingredients of those components in natural products, right? That's ion channel signaling. So next time you put on Tabasco, your trip channels are telling you how much capsaicin is there. And of course, it's important for cancer because cancer cells use these to proliferate. And animal health. Turns out a lot of bugs and parasites can be knocked down or killed because their ion channels are different than the vertebrate ones. All right. So, and you know that these are important because all the mutations that lead to channelopathies, for example, inborn errors of ion channel function cause childhood epilepsy. And a lot of these children that have aberrant activity of ion channels, they don't live past 3 to 5 years old. They're in seizures all the time because of aberrant ion channel activity. Sometimes you have muscle paralysis. Sorry, is that better? All right. I hear an echo, but if that's okay. So you have muscle paralysis due to ion channel mutations. I had a colleague whose heart stopped. He went into sudden cardiac death because he had a mutation in the proteins he studied his whole life. All right. His father and his uncle had the same mutation. And you would think that he would know better to take care of himself. And this was a defect in the delayed rectifier potassium channel in the heart, which we'll talk about. And there's diabetes. There's all kinds of other things. There's even a great story about cystic fibrosis that Vertex does that I'm going to talk about, which is an ion channel mutation. It's a rare disease. The children die because the fluidity of the airway is compromised, the mucus because of chloride, loss of chloride channel activity. Mucus accumulates, and they made drugs that actually have cured this, and now they're actually working on gene replacement. And a Google Award was just given for that. All right, so, and this is why I went to Merck. They have therapeutic utility in all kinds of areas. Blood pressure lowering, for example. Merck forgot through some scientific discussions that they didn't think that calcium channel blockers were very important for a cardiovascular company, but we were able to get the second generation felodipine, which we were able to register, and it is still used as an antihypertensive. It is still it's a generic, but it's best in class. So you can lower blood pressure. Some of you may have atrial fibrillation, and maybe you're taking amiodarone. That's an ion channel blocker, okay? Sometimes people with diabetes used to take a drug that actually stimulated insulin secretion that was found by accident because it came from a bunch of antibiotic sulfate drugs, and all it did was lower glucose in rabbits. And that was glyburide, first real therapy after metformin for type 2 diabetes. I talked about cystic fibrosis, and there's a beautiful story about ivermectin at Merck I'm going to tell you, which is another therapeutic activity. Somebody mentioned to me about the central nervous system. The whole central nervous system is driven by ion channels, all the electrical signaling. We were talking about that. You have anti-epileptics, you have antidepressants, Librium and Valium work through ion channels. Pain, lidocaine is an old pain drug that was synthesized the year I was born, 1949, and is still being used. It's a sodium channel blocker. And there are other indications, like antivirals. There's some influenza drugs. Influenza uses a proton channel in its life cycle, and ranitidine, I believe, is one of those flu drugs that works by blocking an ion channel. Okay, so it's not just mammals we're talking about. This is the heart slide. So the way ion channels work is you put in a complement of ion channels in a cell that's well polarized, and there's a duty cycle of changing membrane potential called an action potential. So in a cell that's sitting at -70, most of the ion channels are closed. If the cell drifts and starts to depolarize, the sodium, this looking at a action potential at the top, but the same thing is recapitulated at the bottom on the nerve action potential. Look at the time frame, though. You would think people think much faster than their heartbeats, because look at the time frame here for finding an action potential. It's not always true. 200 milliseconds up there for a typical cardiac action potential. So the cardiac cell starts to depolarize, sodium channels open. See the upstroke, the depolarization of the membrane, that opens calcium channels, so calcium come in for the muscle contraction. But you can't have the calcium come in all the time, right? You're going to get rigor. So you have to repolarize the cell. So that depolarization opens potassium channels, and you see the the cell repolarizes to an after hyperpolarization, and there's a potassium channel that maintains that resting potential before another cardiac action potential. So it's the working together of a sodium channel, a calcium channel, and potassium channels that allows your heart to beat, okay? And allows your neuron to fire, because down here is the same thing with different ion channels in a different time frame. So the sodium channels will fire, and you get a rapid depolarization, and they shut off because you can't depolarize too long. I mean, nature has taken a very fundamental principle, used a few proteins to just build systems which are so versatile that you can go from cell to cell by just changing the complement of the ion channels. And it's the same thing. We're firing action potentials all the time. It could be in the brain, could be in the heart, could be for calcium ion movement in the kidney. Okay. And so this is how a nerve fires. Sodium channels open, calcium channels open, calcium comes in for the neurotransmitter release, and then you get repolarization, right? This is something we all care about at our age, the cardiac action potential. You go to the cardiologist, and he does an EKG. He ever explained to you what that EKG is? No, of course not. He probably doesn't know he knows from a clinical point of view, but he probably doesn't know in terms of electrical conduction over the heart. So what we have here is we have a heart, and nature said, I need to build a machine that can pump blood for 70, 80, 90 years, okay, in a synchronous fashion. And I'll use what I have, what ion channels I know about, to build different regions of the heart with different channels so I can have an electrical movement from the SA nodes, I know atrial node at the top, over the atrial cells into the ventricle, and then truncate, okay, repolarize. So what an EKG is looking at the cardiac cells. It's a summation of electrical activity of all the different regions of the heart. So when the blood comes into the right atrium and you get contraction to push the blood down into the ventricle and then out to the lungs to get reoxygenated, and it comes back into the atrium and then down to the other ventricle and out through the aorta. Nature has built a machine which is so synchronous, and the summation of all the action potentials of every region of the heart is the EKG. So when the doctor says, oh, you got a PR interval increase, he knows, because if you look at where the PR interval is, it's that upstroke, it's a sodium channel problem. Or he looks at the QRS, which I have a left bundle branch block, all right? And I have a prolonged QRS interval. And I'm telling him what the basis is of it. And they love that. And they really love the next appointment, because I'll bring the paper in to show them. All right. So the QRS is actually that part of the action potentials, which reflects the electrical movement into the ventricle. And then the big killer, the sudden cardiac death, is when you have a QTC. That I'll show you in the next slide, elongation, because that puts this a heart that's doing this into a heart that's doing this, ventricular fibrillation. And if you don't put the paddles on, that's it. Okay. So now you know what an EKG is. And so you can, next time you go see your doctor, what's my QRS reading? How many milliseconds? And he'll be impressed. And then he'll say, find another doctor. All right. And I had a cardiologist for 25 years, and he just loved talking about everything but me. So there are mutations like my friend had in that ion channel, which is responsible for repolarization of the cardiac action potential. You see those potassium channels up here, IKR and IKS. If you block those, you stretch that action potential, the QTC interval becomes very large, and you put your heart into ventricular fibrillation. And why is it? Well, that can be a genetic mutation. It's rare, but the problem is drugs cause that inadvertently, okay? Drugs have been removed from the market, like trifenadine and cisapride and estemazole. These are drugs made not against ion channels, against the receptors and other things, but people were dropping dead, all right? And unknown origin. And now the FDA, before you register a new drug, you have to show that your drug made against another target does not touch any of these cardiac ion channels, because it could cause lethality. So drugs were not only removed from the market, people tried to make drugs that block these potassium channels, these delayed rectifiers, as antiarrhythmic agents. I didn't think that was based on first principles, I didn't think that was a particularly good idea to prolong the action potential, but Merck did it, MK499, Pfizer did it, it's called dofetilide, Eisai did it, and in clinical safety studies, oh my God, we're causing the heart to go into the fibrillation, and we stopped, all right? But Pfizer didn't, because they found out that atrial fibrillation, which is a different type of cardiac arrhythmia, could be controlled with dofetilide, okay? And so it's got a black box warning and can only be used in the cardiac ICU, right? So quick story about this. I have a friend in South Carolina who had atrial fibrillation, and he had a valve replaced, and he spent 2 weeks in the cardiac ICU, and they were going to take him up to his room finally after 2 weeks, and some rocket scientist decided to give him a dose of dofetilide to stabilize him, so he wouldn't go into AF on his way up. And he died in the elevator. He went into VTVF, ventricular tachycardia, ventricular fibrillation. And luckily it was a 3-story hospital, and it didn't stop. And when they opened the door on the 3rd floor, they defibrillated him, all right? Got his rhythm back. And he was only dead for 3 minutes. But And he calls me in February to celebrate the anniversary of his first death. And that is actually a true story, okay? So you have to be careful with your drugs, and that should not, that's why it says use only in the cardiac ICU on the box. All right, so what do these things look like? They're really only 400 genes make up the ion channel family, all right? And nature's really clever because it says, all right, I'm going to use the same principle over and over and over again in every different tissue, and I'm going to use the same basis and just alter it a little bit so I get what I need, okay? So on the left side, you see the voltage-gated superfamily. These are ion channels are just controlled by the membrane potential. You change the voltage of the membrane, they open, they inactivate, whatever. On the right-hand side, you find the ligand-gated superfamilies, all right? That means acetylcholine or GABA-aminobutyric acid or glutamate. Those are ligands that open the channel instead of voltage. It's very simple, all right? And they're all characterized by the ion selectivity. They either move chloride or calcium or maybe very selective for sodium and potassium, which are different sizes. And that's basically it. A few of them move protons. And then the gating, that is how they're open and the voltage dependence and the kinetics. You can build all the action potentials you want just with those three characteristics, and you have the ion selectivity. Now, I will tell you that down here we have some strange ion channels. Right? They don't fit the standard class I've been telling you about. For example, the CFTR that cause cystic fibrosis. That's a chloride channel that Vertex actually was able to drug. That's an outlier. There are some mechanicals, you stretch your skin, you can turn on ion channels because they sense mechanically the movement of the stretching. There's the one I wanted to point out was ENaC on there. That's an epithelial sodium channel that Merck made a drug against. Amiloride is a drug that's used as a diuretic. And it was done in the 1960s, before ion channels were ever described. They used a urine output assay to put compounds in animals and screen. We call it the rat piss assay, okay? It's how much urine comes out if you feed these substitute pyrazines. Ted Craig worked on this. And it turned out they had made a drug that was so potent, nanomolar potency, that the basic researchers were able to use that drug to pull out the first ENaC channel and characterize it. So it's a Merck drug made in a urine output assay that led to the discovery of this ion channel as a new class of ion channels. And that was done in academia. So these things are pretty important, these drugs. Okay, so I put this in here before, and I want to make a comment about this. Basically, the voltage-gated ion superfamily is described on this plot. They start with two pore, that means a pore with two crossings of the transmembrane with the protein. That's a bacterial channel, okay? That's at the bottom here. And then nature adds onto it. It adds more transmembrane crossings, so you have voltage sensitivity. You have calcium sensitivity, builds up a little bit more. You have sensitivity to cyclic nucleotides. It's basically starts from that little thing down there. Sometimes you duplicate it and you get two pore channels, which if you go to the hospital and they give you gas to put you to sleep, that's the target of the action of the anesthetics. And basically, you take a potassium channel, and you multiply it into 4 regions, and you get a sodium channel or a calcium channel. So you start with something small, you build a subunit, and there'll be 4 of these, as I'll show you, for potassium in a complex, and then you extend it, and you make sodium and calcium. Its nature is very clever. It will not reinvent. Once it works, from bacteria to man, it's the same process. And I wanted to mention, if you see the review, I took this from Bill Kaczorowski, an old friend that started this field. He passed away last Wednesday, and I've been friends with him. Family's been friends with him for 40 years because Dr. Garcia purified one of the ion channels. Only a few have ever, she actually purified it from native tissue, and he's the first one to do the sodium and calcium channel purification. So those of us who are into membrane biochemistry. Bill is like the father of the field, and we lost him last week, and I wanted to mention that. I mean, he really did a lot to build and helped us build the field. So this is what these things look like and schematically. And you see, calcium and sodium channels are there, built out of a potassium channel, and you have the two pore. I mean, so what do they look like? So the first structure, and these are simple. This is the first structure of a potassium channel from a bacterium that Rod McKinnon won the Nobel Prize in chemistry for. We know Rod from the old days when he was a postdoc. He actually worked at Merck in our labs for a week or so with Maria doing a collaborative project on it. I'll show you the peptide they purified, which these peptides that I'm going to talk about did a lot to help this, with structural studies. But basically, you look at a top view, you see the hole, see the ions in the hole, you look at a side view, and then strip the side view down. So this is like a very small pore made up of 3 amino acids in 8 times, because it's 4 subunits, right? And 2 crossings. And then the carbonyls of the amino acids, a glycine and tyrosine and glycine, which basically, you see what the pore looks like down there? So you strip the water off of a potassium ion, and it goes to a binding site. And then the next binding site, remember, potassium is high inside the cell and it's low outside. So when you have ions lining up in the pore, the electrostatic repulsion just fires those out into the outside like a machine gun from electrostatic repulsion. The ions are stripped of water, so they're flying out of diffusion control. So you have a pool inside the cell and you're shooting positive charge out, and that's how you get the negative potential in the membrane, right? I mean, this is like elegant. If you want to do things fast, this is how nature built it. And you say, oh, so he did a bacterial channel, but it's a bacterium. It's not the, right. So then he did a mammalian channel, which is KV 1.2. We worked on KV 1.3 as an immunology target. I'll tell you about it at the end. So this looks very much like the bacterial channel, except instead of 2 transmembrane crossings, we have 6. But we have the same pore. And you can see it looks, nature does this the same. It might be 4 subunits, sometimes 5, sometimes 2, but it's always the same concept over and over again. And then, very importantly, you have a beta subunit, which changes the biophysical properties and the pharmacological properties sometimes of ion channels. All right, so this is why I came to talk to you about modulators, because we all need ion channel modulators. If you look at the indications of the drugs that can be developed for the various pathophysiological states that elderly people like me have, for example, hypertension, diabetes, sometimes diarrhea, sometimes not. Thank God, no cystic fibrosis, but deafness, anxiety, pain, epilepsy sometimes. Importantly, look at the bottom. Atrial fibrillation is the most important cardiovascular target that's not treated well these days, and that's all an ion channel driven mechanism, or congestive heart failure. So ion channel diseases are prevalent, and we all suffer as we get older from the expression of aberrant activity in ion channels, or maybe you can correct the disease by targeting an ion channel. So there's an opportunity for drug discovery. And you know, if we understand what the target is and we're not targeting a black box, there's, and we have, and I'll tell you in the last 25 years, it has now become possible to make drugs on this target. So basically, how you want to modulate an ion channel is not how nature does it, because nature with venomous organisms target to kill the prey. You want to do it therapeutically. So we are lucky that ion channels sit in a membrane and go through different conformational states. If you want to block the pore, which is the entry exit, like many of the toxins I'll talk about next do, that's a poison. But if you can target one conformational state with a drug, you can actually interrupt the duty cycle of the ion channel. So for example, if I have a sodium channel that's firing aberrantly, very, very fast, causing a pain signal, if I can block that sodium channel with a drug, not tetrodotoxin, which just plugs the pore, but a drug that selectively recognizes one conformational state, I could get what's called the therapeutic index, because normal firing will not be because of kinetics of drug binding and unbinding, will not be affected, but aberrant activity will. So I'm trying to target aberrant ion channel activity and make a drug, and that's possible. So let me tell you how nature did this first. You all have heard of tetrodotoxin, maybe. It's found in a Japanese fish, fugu fish. Right? It turns out it's a bacteria, makes this alkaloid, lives in the gills of the fish. Now, back in the day in Japan, I mean, people like to eat the fugu fish. I've had it because when you eat it, if there's a little tetrodotoxin contamination, your tongue tingles, okay? But if there's too much, you stop breathing. So back in the day, the Japanese sushi restaurants in Tokyo had an iron lung because the half-life of tetrodotoxin in the blood is less than an hour. So if you take in tetrodotoxin and you can't breathe because you paralyze your diaphragm because you can't fire your sodium channels, put you in the iron lung and 2 hours later you go home. All right. That's a true story. And they when they have fugu fish in New York, it's special in spring. They have a month. They don't have any iron lungs. So they learn to actually, sometimes they heat a little bit the fish, which tetrodotoxin is very heat unstable. That's how they get rid of it. Anyway, you sang about bees, right? In your songs, it's a bee. Apomin, a bee sting. Apomin is a small peptide, brain penetrant. If some people get a bee sting and they die because that's a blocker of a calcium activated K channel in the brain. We have, you see the cone snails in the middle? Cone snails actually have toxins that they project. There'll be a fish and they sting the fish and the fish can't, because it can't, poison its ion channels, and then it dies and then they ingest the fish. There's a drug for pain that comes out of the conotoxin snail, all right? It's called Preout. That actually blocks the Cav 2.2 calcium channel in the spinal cord for pain transmission up to the brain. It has to be given intrathecally. It's usually for cancer patients, but it gets into the brain and has all kinds of side effects. It just shows you a small molecule is better than these peptides. And so we worked a lot with things from spiders and scorpions. That scorpion at the bottom, which is found in the desert in Israel, provided more peptide toxins for researchers. It's Laeris quinquestratus var hebraeus is the name of the scorpion. It provided a lot of probes for people to study ion channels. So the story is that this thing called charybditoxin was purified in an academic lab at Brandeis and given to the community. And it turned out to be a mixture of peptides that block different K channels. So it wasn't pure, but it blocked one of the channels we studied at Merck. So we got the venom and we purified things at Merck. We don't have mixtures. We purified the active component and found out, well, the one we were interested in was one of the there was one peptide there. There was another peptide and another peptide that blocked another one and some sodium channel modulator. Elizabeth Sugg made the charybditoxin. We always make things synthetically at Merck, and that became the standard for the community. We gave out a lot of it, but Rod McKinnon came because there was a contaminant in there that blocked a Drosophila potassium channel that mutations cause shaking. All right, it's called shaker toxin, agitoxin 2. And he needed that peptide for his structural work, and nobody could get it out. But it was in there, all right? You just have to have really good analytical means of separating things. So Maria and Rod published a paper, work he did, they pulled it out, and here's what these things look like working. This is Yang Do Gao did a modeling study. That's the small peptide. All right, it actually fits on top of the ion channel with that lysine, a positive charge, projecting down. So where the potassium binds is where that positive charge binds. It's just a plug. And we actually purified that, and that's the model of it. And then somebody actually did the structure with the real charybditoxin. It wasn't us, many years after. You see, that small peptide, which was used a lot for proof of concept studies. If you think you got a new drug target and you don't have a drug to prove that it's a target, you use one of these for proof of concept. And you can do it in cells and T lymphocytes. You can do it in mini pigs, which we did. I mean, for an immunology program, or you can do it in CNS. These are a cheap way to get proof of concept that, because it's going to take a long time to make to screen and find the ion channel drug. If you have this, you can start. And that's why we're known for these peptides, because they were very important as tools for us. And so basically, you make a drug by having a state-dependent pore blocker. Because, as I say, these ion channels go through all kinds of machinations. You want to target one conformation and spare the other ones. All right? So, and this is the story I want to tell you. All this really interesting science and, God, and this is the calcium channel blockers that Merck first missed the class of nifedipine. Well, how is this discovered? Because this actually opened the whole field, because it bound so tightly to the calcium channels, you could radio label it and then pull out the protein, solubilize, purify the calcium channel, sequence it, clone it. Bill Catterall did a lot of that work. And it was because this very potent thing that the Bayer chemist had discovered. Well, this must be a wonderful story, right, for pharma. So it turns out that these dihydropyridines are made by a simple condensation reaction called the Hench reaction. Okay, I think that's what it's called. And there was a medicinal chemist at Bayer that liked to make these. He was making, I don't know what project he worked on, but it wasn't for his project, because they're easy to make. And Bayer actually made 100,000 of these in the end. He had a brother-in-law who was a pharmacologist. And he said, he gave him the drugs to his brother-in-law. I said, put them in the dog. What did they do? They don't do anything. Here, try again. He kept giving him different analogs. And he said, come on, because they're running. Bayer actually almost fired these guys for doing this. And then they killed the program. And then they made $1 billion drug off the program. And then the guy should have sainthood, but I think they were fired anyway in the end. Anyway, all it did was lower blood pressure in the dog. All right? And it was, as he made these things, it got better and better and better. And so what the field thought, or what some of the safety people at Merck thought, was, how can you block a calcium channel? All right? That's like, calcium is the ion that controls everything inside, it's for contraction and secretion. You can't do this. But it turns out, and this is just and this is where the drug binds. Maria shows the slide out to medicinal chemists after all this lore about you can't make ion channel drugs and all. It actually binds to one specific state of the calcium channel, the inactivated state, which in a cardiac duty cycle, as I'm showing you the action potentials, or in the nerve, which are calcium channel dependent action potentials, so sodium, right? That state is not very prevalent. So you don't block, unless you put in high concentrate, you don't touch the heart, you don't touch the brain. But in your vasculature, where these calcium channels are, and they're sitting at depolarized potential, and they're inactivated state, and they're leaking calcium all the time that's what causes diastolic hypertension. They're contracting the smooth muscle. And so these drugs are just perfect for that state of the calcium channel to cause vasal relaxation and lower high blood pressure. And if it wasn't for what if the guy's brother-in-law was a bacterial guy? I mean, we would not have these. I mean, it is really a story about serendipity, but it tells you an important safety issue, right? You can drug these ion channels if you know what you're doing about the biophysics and the turnover, right? It's just like any other protein. All right, I'll finish. How do we make ion channels the modern way? Well, that's what Roy Vagelos wanted. But Merck, before I got there, had discovered in an assay where they put bed bugs on a rodent and fed them fermentation extracts. And the score was the mouse lives and the bed bug dies. All right, that's the score. And that's how this was found. The ivermectin is a natural product from a bacterium that grows from a golf course in Japan, is where it was isolated. Bacteria makes a strange looking macrolide. And it is a very, very, very potent parasitic agent and kills heartworm, and you use it HeartGuard in your dog, and cat works in sheep for ticks and things like that. It's really a wonderful drug, but it had a human health application that nobody realized, that in the third world, a lot of parasitic infections could be treated by a single dose a year to block the cycle of the parasite. So this was actually given Roy Vagelos and Jimmy Carter, the World Health Organization went, and this drug was given for human health, and it won the Nobel Prize in Medicine. Bill Campbell from Merck, whose office was down the hall from me, was one of the recipients of the 2015 Nobel Prize in Medicine, because this was as a really important ion channel drug. Nobody knew how it worked when it was discovered, but it has human applications, right? And that's a beautiful story. And the Nobel Prize is really for an ion channel modulator. I like to tell my medicinal chemistry friends who say you can't make ion channel drugs. All right. And just so this is more for Matt. This is the first paper where this was published. I know a lot of people on this in the title. I worked Jack Shabala, who's the chemist who actually was able to take that top ring and reduce it so it would be stable and have commercial value. He was in the lab next to me at MIT in the basement of the chemistry building back in the days when we were both graduate students. So he did that piece of it. Helmut Morosic. Look, I mean, Byron Aronson, the NMR guy, he's 101 years old right now. I can't even read it from here. George Albert Schoenberg, who's the natural product chemistry head. I see at the Philharmonic all the time. I mean, and Mike Fisher, I worked with, who's deceased. Even Dick Tolman, I like. He's the last author here. All right. So that's how we did it at Merck before I came. And then I said, well, we can't do that unless. So Roy had given a talk at MIT in '74, I went to, for an advanced biochemistry course. And he said, I'm going to Merck. We're going to do mechanism-based drug discovery. No more voodoo medicine. No more phenotypic screens. Look at that. Look what it did. Phenotypic screen. We're going to do mechanism-based drug discovery. And that's what we tried to do. So what you need is you need to be able to screen a lot of compounds to find a lead that's potent, has selectivity. You're asking for a lot. You're going to the Merck library and interrogating it, 3 million compounds quickly to find a needle in a haystack. Can this be done? Well, you need instrumentation to do this. And all this got built while I was at Merck. I didn't build it. We did help build several of the devices that two of the patch clamps, the Q patch and the Patch Express, we helped with, commercialize, but we used all these things. The last person on the right, Roto Patch, that's his nickname. He did manual patch clamping, which is quite tedious. He's now the vice president of the people that Nanion that make the patch liner. He's doing pretty well. They have machines that work 356 wells. I mean, I'll tell you what this is about in a minute. But one last thing. I had a summer student, Ivy Dick, who joined my department, one of 67 I had, who became an associate and she worked and she built the Patch Express. She's the one who actually did a lot of the experiments. Then she went to Johns Hopkins, got her PhD in biophysics, and now she's a faculty member at University of Maryland. She just won a Paul Cranefield Award, which in physiology, he was the editor of Journal of General Physiology for 30 years. That's a Young Investigator Award for doing Timothy syndrome, which is a calcium channel mutation. Wow, that's like, I'm the grandfather of that, right? And I'm really proud of Ivy. So the people in it's not me, it's the people in my department, worked all these instruments to get where we are today. But this was the key, Nobel Prize for how to do electrophysiology. It's called patch clamping. Now, if you don't remember anything, remember this, because sometimes one thing can change a whole field in terms of technology. I came to Merck and said I wanted to do ion channels. And then I said, I can't do ion channels. This is stupid. You know, I was learning to do sharp electrodes up at marine biology laboratories at Woods Hole, sharp electrodes with that's how they used to do electrophysiology, 2% rate. I spent my second summer up there with my cells trying to record. And that summer, Patch Clamp was published. And that is the ability to go down onto a cell and make a very tight seal. And so you can actually study this, it's a gigaohm seal. And you can actually study the ion channels under the seal, opening and closing the square waves. But if you blow out the if you blow out the patch, you can voltage clamp the whole cell. And so now you can automate it. So you can do 96. What used to take, we could maybe profile 2 drugs a week manually. 96 well and 384 and 1536 automated patch clamping. Now, like and they're talking about 3, 4, 5, 6. All right. So that is really, that changed everything. But that's like a very mechanistic intensive thing. You can study the conformational states to get a lot of data out of it. You need to look at 3 million compounds in 6 weeks. All right. So another Nobel Prize given to Roger Chen, who was at San Diego, for all these dyes he developed for monitoring calcium inside or membrane potential, fluorescent proteins. That's what he won the prize for. One of his dyes, that senses the membrane potential across the cell membrane. I can tell you how it works. It's a fret-based fluorescence assay that's, you can put it in 3,456 well format. It's that, and it's simple because when you have a cell that's well polarized and you block the channel that's polarizing it, it goes to 0. So what you have, I mean, anybody can do this, even me. If you have a cell without your ion channel and you look at the dyes, that dye, which senses where the potential, if there's no potential in the cell, it's just flopping around that the second dye in there. And there's no fluorescence, resonance, energy transfer. As soon as you hyperpolarize the cell, that dye, which is negatively charged, goes off the inner membrane, goes to the top membrane, and it talks to the other dye that's covalently labeled. All right. And so you get a beautiful signal when you depolarize with potassium. You see that big control? I could see that. If you had pre-treated the cells with an inhibitor, however, and you hit with potassium, it's that. You lost the signal. All you have to do is look at a plate and say, oh, that's an inhibitor. That's an inhibitor. I mean, you don't, if you build the assay right and you do the experiment, well, anybody could do this. And everybody does now. We were one of the first to do that. Then the last technique you have to know, sometimes you don't want to measure membrane potential. You want to look at the ion itself. And it turns out that thallium is a surrogate for potassium. Thallium will go through potassium channels and there are dyes for thallium. So you just measure a thallium signal, right? Now, why thallium? Well, thallium, you know, the safety guys came around the lab when we started using thallium because it's really a toxic compound. There are situations where medicinal chemists have killed their spouses by putting thallium in their tea and things like that. And in New Jersey, actually, this is a poisonous compound, and we used it freely. Safety guys said, we use gloves, and it's not a problem. This revolutionized things. Okay, and last data slide, I guess. Oh, 2 more. All right, so this is just one example of what we were able to do at Merck. We were able to find one compound out of the entire 3,000 compound sample collection, which was a very potent I think 0.6 nanomolar inhibitor out of the box of the channel we were interested in. And the chemists looked at that and said, oh, we don't like it. It's an acylguanidine. I don't know. But a couple other chemists outside, 2 actually, took it, made 100 compounds and made it chemically tractable so that we had to actually brought it back in and had a clinical development candidate in a year and a half, which is, and in fact, we were able to make the most potent ion channel blockers I've ever seen in the literature. This is 60 femtomolar, picomolar, excuse me, 60 picomolar. Ivermectin is femtomolar. Picomolar, that's very tight binding, right? And actually the drug binding site's been mapped by Bayer now in the channel because it's an insecticide target. It just shows you the power of the screening technology, right? But then sometimes you work with Matt Wyrath on a KV1.3 channel, which is one of a member of 7 members of the family, and they're all so closely related, like 98% homologous, and you say, that's undruggable. I can't make a drug. Well, you made those peptides. Why don't you? And I'll tell you why we don't use those pore blocking peptides from the scorpion for this. And he's right, because you cannot find a space which is unique enough to drug, but nature can. And what people have been doing now, especially on the KV1.3 channel target we worked on in the old days, they make antibodies now to the native protein that block function. So they let nature make an antibody that will bind and block the activity of the channel. And you can do this in 6 months, not 3 years. I mean, you can be at a clinic in a year instead of 5. No DMP, no drug metabolism, pharmacokinetic problems with the antibody. And I had 4 clients that did this independently. They took the Kv1.3 native channel and they made antibodies that were nanomolar potency that had 5,000 or more selectivity between the neighboring channels. We got 10 if we were lucky. And you don't want to block the neighboring channels because you get CNS toxicity. So nature, this is the new arm. And my clients that I consult for do this. And the last thing is, well, what happened to those pore blocking peptides that the scorpion made? Well, pharma did that. One company did that. You know what happens when you have little peptides like that have charges on them? The immune system reacts to them and they neutralize those peptides. So they start working in men real well for a couple of weeks, and then because you got neutralizing antibodies. So those things from nature are no good, but use the antibodies against it, the target, and it works. All right. So I've told you a lot about ion channels. The important thing is they're also not only big therapeutic targets, but there are a lot of rare diseases that children have that are due to ion channel abnormalities. And it is now possible because of the human genetics and validation for smaller groups to really try to do this and do it for the patients. Right. And the fact that now people are doing it more rationally, like Vertex, which is organized by an ex-Merck medicinal chemist, Josh Bozier. They have sodium channel blockers in phase 3 for pain in the clinic. They've got some APOL1 inhibitors for kidney disease in Black population. And there's Xenon, which Charlie Cohn went and led the effort on, has an anti-epileptic drug that's in phase 3 for going for registration. So that's the end. And I hope I haven't bored you too much, but it's just a hole with a physiological role. So. Stay right up here. Oh, that's Q&A, which I will moderate. All right.
Well, so first thing, everybody take a slow, deep breath. And then, oh, this is amazing. I was wondering whether anybody would be able to formulate a good question. It looks like we have candidates who can. So let's get started. Mano, come right up and ask your question. No, come up here. No, Mano, here. Now, for the people on Zoom, our speaker is sitting down off camera. So you will hear him, but you will not see him. You will see the people asking the questions. Thank you.
Well, good morning, and thank you very much for a very interesting, I think, and a complicated story. I wouldn't want to bet on how many of the people out there really understood what you were talking about.
But they will not be bitten by bees, I bet.
Oh, yes. So my question is this, that all this new studies that you're involved with, have been involved with, and so on, and you've brought new science to the world. And I think it's going to continue happening over the years if we don't blow ourselves up in the meantime. So my question is that as nature was developing mankind, and as we were, becoming from fish and whatever we were before and became the humankind, why was that development so complicated, which you're describing now? That's what's happening in our bodies and so on. Why did nature make it into such a complicated situation that you're still studying it now, millions of years later? I don't know if there's an answer for that, but I'm very curious about that.
Well, you use the calculator. Closer to your mouth. Like this? Yeah. So you used a calculator in the past, and then you use a computer now, right? So what nature did, this took a long time. This is an evolution. This is a great example of evolutionary development, right? Because the channel proteins that I talk about started in simple organisms. They only moved maybe one class of ions, right? but they were important for the cell to live, just the cell, like the bacterial cell, right? And it was all bioenergetics, actually, that Peter Mitchell got his Nobel Prize for. How do you energize a cell so that it has enough power to keep growing and then reproduce? And that's really the key. The chemiosmotic hypothesis was the first key here for what I've been talking about, the biophysics. So nature did that first in small organisms. And then as it built more larger organisms, for example, the transition from the sea to land was the kidney. You know, there's other things that out there that don't have kidneys because they don't have, the salt in the ocean, they live in, right? The homeostasis of salt handling in their body is appropriate for the sea, but we don't live in the sea, so we have to control our salts, right? So we have kidneys. So you can see in the evolution of various, as the organisms got more complex, this is basically the same principle used over and over and over again, made more complicated. But you think about the brain. I mean, that is all ion channel driven. It's all ion channel driven. Okay. All this electrical signaling, maybe even the storage. All right. All the neural circuitries, which are different. I've never been clever enough to study the brain, so I don't do neurobiology, right? Because it is really common. But the heart, think of the heart. Your heart beats how many times in your lifetime?
Oh,
right.
times.
And if it wasn't for the asynchronous activity over the heart, different heart cells. You know who worked out the heart, how it was beating? Electrical engineers worked it out, okay? In England. Electrical engineers were the two Chen brothers, Dick Chen and Roger Chen. Roger won the Nobel Prize for those dice. They were double E's at MIT as undergraduates. They went to England and worked with people who did modeling of electrical. A lot of this comes from electrical engineers backwards to biology. But if we didn't have it, we wouldn't be here.
Well, that's the thing. What happened there, that it happened from the fish and so on, that all these developments, all these different mistakes that you're not correcting with medications.
Well, so why do you have these diseases? Well, for example, you get a mutation on something that you don't want to mutate. I mean, that's why those people die. It's natural selection. They're not meant to. If you have them, you select for the fittest, right? So the ion channel evolution is because of fitness of physiology, right? And all the accidents go away, right? Now we try to correct. Why do you have hypertension as you get older? Well, it's probably salt and things but people didn't have that when they were young, probably. And it's accumulation of a lot of other factors, right? But you have to correct an aberrant activity, which is not there when you're young. Your blood pressure is normal, right? So people said, well, we'll target the things that are controlling the pathophysiology. So, and how did it get done? Charles Darwin had an idea, and I don't have anything better to give you. But if you think the computers are good these days, nature made the best computer. There's nothing better. And it's all ion channel based. I'm happy to say, I didn't know what this was in high school. We talked about the genetic code when I was young, right? I mean, and people didn't even believe that these were proteins. They thought that the membrane opened and closed transiently and magically. I mean, nobody, until they actually purified the proteins, and that only was 1980. This is not an old field. It's only 45 years.
Thank you.
All right. It's not very satisfactory, but it's the best I could do.
Well, now that question was from a doctor. Now let's hear from somebody who's not a doctor. Paul, come on up.
These two are.
Oh, Mitch, okay.
We got the wrong order on you. Hey, thank you so much for explaining this. Now, you and I are about the same age, and when we were teenagers, I was looking at a globe, and I looked at the Atlantic Ocean, and I saw Africa, Europe, and the Americas, and said, they like fit together. So I went up to the teacher and I said, how come they fit together? And she said, I said, there must be something where the continents pulled apart. And she said, no, that's just coincidence. And lo and behold, it'd been known since 1915, but it never got around to, educational level until the 70s. So the question is, this isn't a geology thing, it's just a story. So my question is, how did they explain all this stuff before you invented ion channels?
Well, I didn't invent it. We discovered them, right? I mean, so that's the point. How did they, it was all electrical modeling. So they would make like going back into the 20th century, Katz and Milady won a Nobel Prize for taking a squid axon and actually measuring electrical impulses that go down the axon, right? It's the basis of neuroconduction. You know, everything going on here. This is, I don't know, late 1950s, right? And that was revolutionary. But what was causing that? You know, what was it's an electrical signal we can measure. We know what ions are being used for the signaling, but how does it happen? You know, I was up at Woods Hole in '81, and they used to have what they called the Tuesday night fights, which was professors came in and gave a chalk talk, right? And all the biophysicists, postdocs, I'm not one of them, but I was invited to join to learn electrophysiology. And they would argue about, and then this guy got up from University of Pennsylvania, and he said, a channel has to look like this. And he drew basically what I'm talking about. And he said, because I can do this experiment in a squid axon where I take pronase, which is a protease. It just chews up protein. And I can infuse that pronase inside that squid axon. And all of a sudden now, the sodium channels, which used to go inactivate, up, open, close, well, the rested, open, and activate very quickly. That's otherwise it's toxic. There's a confirmation, very fast transition. He lost that transition with the pronase. So he said, this has to be a protein, not some hole opening up, you know. And I said to John Rubin, who's my mentor from Columbia, who taught me electrophysiology, actually, we learned it together with the patch clamp. I said, what's this guy smoking? Yeah, well,
All right.
Turns out he was right and I was wrong. And I was stupid and young and I didn't know. Of course, it's got to be protein-based, but until Bill Kaczorowski pulled out the first sodium channel, all right, then we had a protein. As soon as we have proteins, we could make models because they're just like enzymes, but they're in the oil, they're in the grease, and they open and close instead of, that's their catalysis.
We'll have the couple of people from online. So Bill Tittle, please. Come up.
Yeah.
A little bit of
So Greg, thank you very much for that. It's great to see a guy that's passionate about something, and you obviously I have this feeling like I just sat through a 15-week course in an hour. So if I want to, I took out my 1,000-page biochemistry book, and they do have a section on ion channels, which I'm very glad to do, because I just so my question is, could you repeat again, what is it that opens an ion channel?
What is it that opens? It's the pore that opens. The pore is shut. Oh, the pore is shut and it opens, right? Because it's a conformational change. The channel sits in the membrane at a closed state. If you can change the membrane potential, which the cell is sitting at -80, and you go to 0, you depolarize the cell. Wow. A protein sitting in 9,000 volt energy field, like in 1 millimeter, when you go from 9,000 volts to 0, that's a big relaxation, right?
Mm-hmm.
And that relaxation is recapitulated in the conformational change that allows the pore to open. And then these ions go through like a bat out of hell, right? They're diffusion control, because you got to have it done like this. It's an instantaneous type thing. And then you got to shut it, right? Because if it's open too long, you'll kill the cell. And that's why sodium channels inactivate, others don't inactivate. Nature figured this out. And you know, that biochemistry book, when I was in school and I used Mahler and Cordes, I used all these books, there was nothing about ion channels in them, right? This is 1980-ish, 82, 83 onward. If they hadn't invented patch clamp, we'd still be doing stupid sharp electrode recordings and we would learn nothing. So it was technology. We had ideas, but you got to have the technology to measure it. And ion channels, the Nobel Prize for patch clamping revolutionized everything. And then the high throughput screening, which is a friend of the medicinal chemist because they don't want to work on anything, just something good. And that's, so I actually am blessed to have been through an evolution of a field that I didn't know existed before. And in fact, that Roy Vagelos would hire someone to come and do this when it had not been done is like a testimony to Roy because he was a visionary.
Thank you very much.
Yeah.
One more. We'll take one more question from online first. So L.L.C, please.
Thank you for a very informative talk. Is it known what role ion channels play in producing the sensation of consciousness in human beings?
Well, I can tell you that the channels that are modulated when you go to sleep before your operation are 2-pore potassium channels. And the way that you go to sleep is actually you depolarize certain neurons. And then if they wash out and you come back. So consciousness has to be involved with obviously the polarization state of, but that makes sense because when neurons are polarized, they have the ability to talk to each other because they're not isolated. They're all coupled. They're all sensing each other's. And it's really the change in membrane potential that alters the talk between neurons. So actually, nobody knew how the anesthetics worked until they found the 2-pore channels, 2-pore potassium channels. And so CNS is all based on ion channel activity. Look at epilepsy for sure, because it's the aberrant activity of an ion channel. And they give a very bad drug called valproic acid, which is a fatty acid. How does that work? It just dampens all the excitability of all the ion channels. So it's, it's just dampening. It's not a therapeutic as much as treats a condition. So while I'm telling you this is all worked out, there's still a lot to do here with CNS, okay? And maybe I'll be around long enough to see it, but yeah, some really smart people work in this area. Unlike the old biochemists that started just, chemists, just kind of simple views of life.
Thank you.
Right, we are running out of time. Next question from Kirk Benz.
This is a very personal question. I had a long bout of chronic Lyme disease, which resulted in what they call heart blockage partial and then heart blockage total, and eventually a pacemaker, which works fine. Was that? Good for you. Is that something to do with ions? It was explained to me in terms of destroying nerves that go to the bottom of the heart.
All right, so I actually believe my AV, I had 3rd-degree AV block that came out of nowhere when I was, last October, I went to a memorial service for my PhD mentor at Stanford. And on the way home, I just went into AV block without any stage 1 or stage 2. And the first thing my doctor looked at was Lyme disease. Did I ever have Lyme disease? And no, I never had it. I tested negative for it. Nobody knows the connection this is a very bad bacteria. And apparently the circuits, it infects the heart. So if you infect the heart, you'll burn out circuits, right? Now, I started from a position which had a left bundle branch block. And that was because of untreated hypertension when I was young. And that's the conduction system in the left ventricle was damaged. And I had that condition for 25, 30 years, and there's nothing you can do for it. It's just, you just live with it. It didn't get any, it's pretty much stable. You have to be qualified for operations. So when this other thing happens, like this AV block, and I think I know what drug caused that is, and I won't, you're recording this, so I'm not going to say what it is. But there has to be a reason. And I actually, from the literature, went and figured it out because of the effect of the other drug I was taking. And so, Lyme disease has to, and I had never heard that it causes heart block. All right, AV, that's the AV node, right? That's the interruption of the signal. And so it must be the infection that hurts the cells, right? And it must be just what tissue distribution of the bacteria, which is hard to get rid of, right? So And did I ever think I was going to have a pacemaker? No, but I get to look at electrical recordings now all the time. Much to my cardiac electrophysiologist, I got this guy called the cardiac electrophysiologist. He walks in, they had the pacemaker put. He said, "I'm a cardiac electrophysiologist." I said, "I am too." He said, "You are?" And I said, I showed him the action potential. Want to talk about this? It's funny. Medicine and science are just really joined at the hip.
Okay, we're good. Unfortunately, we're running out of time. So the last question.
Well, first, let me say that I feel a little bit like Bill Tittle in that I've had a short course in this. And yeah, despite all of the complicated aspects of this, I thought you did a fabulous job of really explaining what was going on without having to know about all those chemicals. I mean, I really feel I understand what you were talking about. So I acknowledge you for that. Now, I have two questions.
One of Dr. Garcia's students.
Yeah.
Makes
Yeah.
you know what they are.
I've heard some of a part of your story because Bill Campbell is a good friend of mine when he was living in Summit. But anyway, two questions. I don't know if there's time to answer one or no, I'll pose them both and you can answer one or the other or neither. And one is this: you've explained the whole thing in terms of a single cell, and yet obviously there must be a whole ensemble of cells, hundreds of thousands, millions of cells, all doing the same thing, having to do the same thing. So how do they all coordinate at this time scale? That's the one question. And the other question is, these all amount to tiny electrical pulses moving around, the ions and so on and so forth. And there's enough of them left over that they go out into the tissues and they reach the surface and we can measure them. So why don't all those electrical pulses interfere with other things going on?
Oh, like your TV. Right. Yeah, that's actually an interesting second question. I never thought of that, but they're very small, the electrical fields that we're generating. But your first question was shown on the heart. So the heart is just not made up of, there are atrial cells, there are ventricle cells, there are pacemaker cells, there are cells that do conduction, and the wave spreads over, for contraction, the electrical wave spreads over the ventricle muscle, causing the pumping, right? And so, and not every one of those cells has the same ion channel complement. You can take a potassium channel and have 50 varieties of it in terms of kinetics. You can also phosphorylate or you can change, because it's a protein, you can change its activity by regulation, phosphorylation, or lipids around it or whatever. And so nature has, it is remarkable, uses in a specialized organ like the heart, has done the architecture so that the cell types change to actually allow the wave to progress, because an atrial cell is different from a ventricular cell, from a Purkinje fiber, from a SA node. We know all that. But nature put it together so it works, okay? And when you have a sudden cardiac, what do? They shock you. They defibrillate you. They actually depolarize and let you repolarize again. They get rid of this ventricular fibrillation by just stopping it and then let it start again. And that sometimes it works, many times it works, sometimes it doesn't. Lucky for my friend in South Carolina, on the third floor they had the paddles, right? Yeah, right. And then the electrical fields, by the way, your Apple Watch, when you do an EKG, it's not really the EKG in the doctor's office, right? It's actually measuring surface potential of your body is what you're looking at. And it's not high quality, but it tells you about rhythmicity, right? Same thing with heart rate. It's not a direct measurement. Thank you.
Thank you so much for this incredibly stimulating talk. Now our MPC, Matt, will thank you especially for it.
Oh, great.
I'd rather have mom friend.
For your time today, we have a certificate of appreciation for you. And you'll notice that our flower here is an orchid.
Yes.
Because when we were founded in 1930, Summit was the orchid capital of the East Coast.
Oh, I didn't know this.
So, yeah.
Oh, very nice.
And we like to also have the Summit salute, the old guard salute.
thank you for all for coming, and thanks to Matt for inviting me, and thank you for Joel for making this work, because I'm not, even though I do electrophysiology, I don't do this kind of stuff. He's a